So welcome everybody to the April Facts webinar, which is now not a Facts webinar, but a COVID-19 webinar, with COVID-19 taking it over just like it's taken over so much else. We have three speakers for you today, Kert, Viele, Scott Berry, Ben Saville, who are going to talk as according to the agenda here. We're going to record the presentation part, and then at the end, to allow the Q&A to be more informal, we'll turn the recording off. So we're just going to hang on a few minutes while the counter on the number to allow the counter on the attendees joining to stop ticking over quite so quickly. So Kert boasted that he'd broadcast out an invitation to this to his 10,000 followers. Yeah, we're going to struggle if they all turn up.
I did no such thing. But
no, we just wait for them, people to finish joining. I'm not sure if we have, if we've kept records for attendee numbers, but I think we might be setting a record already. Oh, it's slowing down. Oh, no, it's not.
It's like watching the disease spread.
only much more pleasant.
Yes.
So, so Kert, as you're off first, and as you've got another appointment shortly after you've finished speaking, do you want to at least take over the screen and get ready?
Sure, I will do that now. I need you to stop sharing.
Okie doke. There we go. So just as Kert gets ready, just to say out loud to everybody, if you've got questions during the presentations, please use the Zoom's Q&A feature. That's what we'll be monitoring if you need to ask any questions. If your question looks like it needs to be addressed immediately, I'll interrupt the speakers and pass your question on. But if it looks like it's something more for discussion at the end, then we'll park it and we'll go through, we'll bring it up at the end in the discussion session.
All right, Tom, should I go ahead?
Take it away, Kert, take it away.
All right, so I'm going to give a brief introduction to COVID-19 trials, kind of lay out the landscape, talk about why we might be interested in platform trials, and how what Scott and Ben are going to talk about kind of fits into the world. Certainly, I do not need to give an introduction to COVID-19. While we are all separate, this is one of the things that really has bound all of us together in the world at the moment. So, and some ironies here, but certainly this is a common problem for us all to solve together. Right now, we're faced with a large number of possible therapies. I've put some down on here that have been in the news lately. There are many, many, many others. And we know that there's a large number of patients that are suffering from this illness. So we would like some way to experiment in a way that allows us to take the large number of people who are suffering, investigate all of these preferably, and figure out what works and what doesn't. And also we would like to do this in the most efficient manner possible because we'd like an answer quickly. Now, one thing that's going on in the world right now is there are lots and lots and lots and lots of small trials. So you can see websites. I just picked a random trials tracker off the internet, and you can see this one has 1,076 trials as of this morning in it. Some of these trials are big trials, they're randomized trials. Some of them are small observational studies, but there are many, many experiments going on. And these are all going on simultaneously. And one question is, well, what is that going to do for us as a society? we'd like to get answers to our questions. We'd like to know, does HCQ work? Does remdesivir work? Does convalescent plasma work? And we run into the problem with all these trials going on, most of them small, many of them observational, that we have an issue where we've got, say, for example, this is a March 18th headline when hydroxychloroquine was being highly touted. So this talks about, remember, there was a very small study in France, non-randomized. Lots of issues have been brought up about this particular trial, but it generated a ton of hype. And if you go forward, these are headlines on April 6th, where there's discussions of anecdotal evidence in New York hospitals. And then we get to April 14th, and we now have some studies that are halting due to safety concerns. This particular one was in Brazil, but there are others in Europe and elsewhere, China, that have been stopped for safety concerns, particularly at the higher doses. And then over the last couple days, we've seen a couple other studies. The VA study, which is, again, not randomized, showed no benefit, higher death rates, and another study coming out from New York that indicated that there wasn't evidence of harm, but there also wasn't evidence of benefit. So we've got all these conflicting studies going on, and small studies, observational studies. Keep in mind that observational studies come with biases. All the data that we've analyzed in-house suggests that observational studies, one group or the other, is a higher risk group, which makes it very hard to interpret these studies. So this isn't really giving us an answer. This is just taking us on a roller coaster where it works one day and it doesn't work the other. Granted, HCQ has been highly politicized, but certainly this happens in other drugs. Remdesivir, we've been on a roller coaster this week where there was a report a week ago out of Chicago that said patients were doing well on remdesivir, and there was leaked data yesterday saying that perhaps there's no benefit. Perhaps the study was inconclusive, it was too small. But anyway, all of these studies coming out where we're either getting partial results or we're getting results from a small number of patients, what we're creating is more confusion than actual answers. So the solution here is to try to do bigger randomized trials. And one of the things we would like to do is run these cooperatively in order to go fast enough. If we've got a lot of people working together, they're all within a common structure, we can therefore run a trial that actually reads out in a short amount of time and we get a more definitive answer. Another issue that's come up that this naturally solves is the notion that we don't want to give too many placebos in the world. If we run 50 randomized trials separately, we are going to find that half of patients in the world are getting placebos. That makes no sense. If we run bigger trials, involve multiple arms, we can limit the number of placebos. And you can see this being done in the UK right now. The headline, they're trying to get patients to enroll in 3 clinical trials. Those are Remap-Cap, Recovery, and Principle. We're talking about Remap-Cap and Principle that we've been involved in. Recovery has a separate webinar. We're not involved in Recovery, but I will give an advertisement. There's another webinar at 1 o'clock for Recovery given by those investigators. The general principles for a trial like this are to explore many arms, put as many things as you can all at once, allows you to compare many things at once, share the control. You'd like to fail arms that don't work fast. If something doesn't work, drop it out, bring in the next thing. This is a situation where most therapies, unfortunately, aren't going to work. We need to explore a lot in order to find answers. We want to adaptively focus on the most promising arms. So we want to be able, if something looks like it's performing better, let's put more subjects on it and try to get it to a readout faster, Or conversely, it may regress back to a poor mean, and we may learn quickly that no, this was an early signal, but it was a spurious signal. So that adaptation works for telling us that things work and telling us that things don't. And then we want to invest our savings forward. So anything we save by eliminating drugs for futility, we want to immediately explore new things. And so platforms achieve these goals. Remap-Cap principle have a platform trial kind of structure. This graph, we've got videos on platforms in general in other locations. But I just want to show this table again. This is from the paper by Ben Saville and Scott Berry. And you can see that if you're trying to investigate multiple arms, the key number here, that 1966, is a notion if you just explore one drug at a time, run a trial, see if it works. If it doesn't, try something else. If that doesn't, try something else, and so on. You can see that this is, in their example, it took about 2,000 patients to find a reasonable answer. Find a drug that works. And this is all proportionally. COVID, I'm not saying it will take 2,000 subjects. As you add features, you investigate multiple drugs at once, you fail drugs that perform poorly, that's the second row, and you invest your savings forward, gradually you can see that sample size go down. And again, think of this as proportionally. So 1966 down to 640, we're talking about tripling or cutting the time by a third in terms of getting to an effective treatment as quickly as possible. So I just wanted to lay the land here. Scott's going to talk about endpoints in addition, which are an extremely important aspect of designing a COVID trial. And then we're going to talk about the details of the trial and how these principles are employed in now the real world in enrolling trials. So let me stop sharing here. And Tom, I'll turn it back to the moderator.
Thank you very much, Kurt. So Scott, yes, well done. He's taken over, and you're up next.
Can you hear me, Tom?
Yeah, we can hear you loud and clear. Thank you, Scott.
And you see my slides?
Yes.
All right. Just a comment as to where we're going on this. Kurt gave a really nice introduction. I'm going to talk about endpoints, analyses of these endpoints. And then going to talk about the principal trial and the Remap-Cap trial as two examples of that. We think this will go just over an hour or so, and then we're happy to have discussions, answer questions, debate different points for the remainder of that hour. So planning a couple hours if people are interested and hang about during that time. My discussion of endpoints are really about what I'm seeing, questions I'm being asked, things we're doing in trials. We being Berry consultants, we're involved in a number of efforts. And so these are different things I'm being asked, different things we've done. I'm not necessarily promoting something here, but presenting discussion. So what's kind of interesting about COVID-19 and the trials is that most of the endpoints are ordinal scales. We as statisticians debate long and hard, how do you analyze ordinal endpoints? What is the appropriate aspect of ordinal endpoints? Kind of interesting that this is somewhat of a controversial is not necessarily the right thing, but debated a lot among statisticians. And here we are where most of the COVID-19 tend to be ordinal endpoints. Most are trying to combine together in an ordinal scale the different clinical states of disease or disease progression and combining those. And it's relatively easy to say this is a worse clinical state than that one. death is worse than mechanical ventilation. Mechanical ventilation is worse than non-invasive ventilation. Non-invasive ventilation is worse than in the hospital but no organ support, and so on. The COVID-19 endpoints are collected over a relatively short timeline. This isn't an oncology trial where you're doing overall survival and you might look at a year or 2 years, median survival of 9 months. These are mostly within a month. Now, within a pandemic, maybe that's not all that short. And so Ben's going to talk about where their endpoint, I think, is about 28, 30 days, and they have to do longitudinal modeling because they're enrolling so quickly and they're doing interim so frequently that you have subjects that are enrolled but not to that time point. But it is generally relatively short. There have been some efforts to try to integrate over time. Is it a single time point that you look at an ordinal scale? Is it averaged? And so there are interesting aspects of how do we deal with time for the subjects. Overall, the endpoint part in most of these trials have been about we need something that's easy to collect, reliable, relatively hard, being objective, and clinically relevant. Within a pandemic setting, resources are short. Research staff have been pulled back out of hospitals. Given the infectious nature of this, it's harder to collect things. So all of this goes into what endpoint should be collected in these trials. It's relevant to think about the disease states. So in most of the trials we've seen, they've been categorized into 4 groups. A severe state are subjects in the ICU, some level of organ support, potentially ventilator, under the care of an intensivist as this severe state. There's a moderate state which tends to be hospitalized, but not yet in the ICU. And ICU is somewhat synonymous with non-organ support, but in the hospital. Then there's a group of mild that are, by the way, all 3 of these top are COVID suspected or proven. The mild tend to be ambulatory, they're in their home, they're non-hospitalized as a group. But all 3 of the top are suspected or proven of COVID-19. The 4th are subjects, participants do not have COVID-19. You might be doing prevention trials. I think we're all going to be talking about the top 3 groups in some form or other. The sort of severity of disease has something to do with the endpoint, of course, is what's clinically relevant for those patients. Some of these, I think, are appropriate across the whole scale. The World Health Organization and the NIH have created ordinal scales that have been adopted by many trials. And you can see this ordinal classification of clinical status. So for the WHO scale, by the way, I think really the difference is that the NIH have lumped together 5 and 6 into 7 outcomes, where there are 8 outcomes on the WHO scale within this. And it ranges across the disease, really, in the clinical states, from death being the worst state, some level of mechanical ventilation, non-invasive ventilation, to hospitalized with oxygen support, hospitalized but not oxygen support, down to non-hospitalized, no symptoms essentially, or limitations, to essentially symptom-free, non-hospitalized. So it's a nice scale across the clinical spectrum of disease, much like the NIH scale. Other endpoints we've seen in trials, mortality as this, and of course it's an ordinal scale. It's just an ordinal scale with 2 outcomes, dead or not dead. We're involved in a trial, anticoagulation trial in Canada, the ATTACK trial, where they have a 3-point ordinal scale: death, mechanical ventilation, or being alive and not mechanical ventilation are the 3 outcomes for that trial. We've seen hospitalized and not hospitalized, and that might be done in a mild population where mortality is really quite low. And the question is, can you prevent hospitalization, essentially a progression of disease? Time to liberation, a time to event endpoint. And of course, as statisticians, ordinal endpoints and time to event endpoints are the same thing. We use very similar methodology on them. They're really ordinal scales with some different level of censoring for them. It might be time to leave the ICU, time off mechanical ventilation, liberation from mechanical ventilation, time to leave a hospital setting can be used within these endpoints. The last one here is one that's in the Remap-Cap trial, and ICU free days, which is a composite of death and time to liberation, essentially, where it's an ordinal endpoint, and the worst outcome is death. We call that a -1, though the numerical value you give it is irrelevant. We could call it a -99, a -10,000. It's just the ordinal value. We could call it A. That's the worst outcome, and that's in-hospital death. 0 means over 21 days they have organ support. That's what ICU, quote unquote, means, is you're under organ support. 1 means that you had 1 day free of organ support over 21 days. And then 2 all the way to 21 means that you never had ICU organ support. ICU is a hard thing to define in this disease, as you might not actually be in an intensive care unit. So in this trial, it's defined as organ support, whether that's given in a field hospital, in an ICU, in the hospital, it's organ support, and that can be really well defined. It's a hard endpoint. And so this is a 23-point scale of ordinal scale. That has the added value that it integrates time and ordinal values. It sort of integrates, first of all, mortality and then support level, but it integrates support level over 21 days. The NIH scale, I think the remdesivir trial, looks at an ordinal endpoint at a single point in time. On day 15, what is your status? It ignores how you got there. You may have been pretty well, you're doing pretty well, and then you jump up to a particular level on day 15. It might be that you had mechanical ventilation for 13 days, you got off of it, you're doing pretty well. The path you got there doesn't matter. It's looked at a single point in time. We've seen efforts to classify the worst value of the NIH scale over 15 days or over 21 days. So if you ended up on ventilation, you end up as the second worst value. If death occurs, it's the worst value. So it's what was the worst value you saw over that 15 days. ICU free days naturally integrates over 21 days. Were you on organ support, yes or no? Over those days, integrated with mortality. I've seen efforts and thoughts, I've never seen it actually implemented yet, to try to do something like an area under the curve, or a progression model over time that tries to look at ordinal values over time. And some back and forth thoughts on that. I haven't seen it actually implemented. Something I think could be fruitful. It has the negative of being quote unquote complicated, which I like, but others don't necessarily like the simplicity of the endpoint. One of the nice things about these ordinal scales is that it's relevant, I think, for the entirety of the disease spectrum. For severe, it's mostly about mortality. Can you prevent mortality? You might have a 50% mortality rate. And then that becomes the relevant part. For moderate, it might be over the entire spectrum or sort of the top quarter, the 3/4 of it, where mild is mostly about avoiding hospitalization. The mortality rate is really quite low, and then the lower part's differentiating. So in many trials, you might be enrolling the entire spectrum, and doing a stratified analysis of the NIH scale is a really relevant thing across the disease spectrum. So we like to avoid different endpoints for different patients. It's awkward in analyzing it and interpreting the trial. And so some of these can be used across the disease spectrum, like ICU-free days, organ support-free days. Again, avoiding organ support at all ends up being the endpoint for mild, where avoiding death tends to be the endpoint for more severe, and moderate can range across the spectrum of disease, which is a nice thing about some of these endpoints. How are these analyzed? And this is what I alluded to, that we argue as statisticians, and it's easy actually to criticize every one of these methods within the disease. You can do a Wilcoxon test, and this is where sort of time to event and ordinal things come together as a non-parametric test. It might be harder to interpret. You don't really get an effect size. You just get a test that these two distributions are different. It's harder to model covariates. It's harder to model different parts. You can do stratified Wilcoxon in those tests. Proportional odds model is, I think, where most of these are going. It's where we've gravitated to. It's relatively easy to model stratification, covariates, time in the trial. You get an odds ratio out of it. The proportional odds model reduces to a logistic regression if you have two outcomes, which is nice. The odds ratio transforms across these scales, saying there's a 1.5 odds ratio is meaningful. It doesn't mean everybody understands it, but it's meaningful. It does have the aspect that every jump in the ordinal scale means relatively the same thing. There's a progression. So you want to be careful that you don't create a scale where tiny little jumps means the same thing as the jump from mechanical ventilation to death, for example, in that scale. I've seen numerical averaging. So you take the NIH scale and you average the 8 values as though they're a numerical value. I hope most people see the issues with this. The numerical average is meaningless in these scales, but yet people are doing it, and they're doing a t-test. Utility analysis of the ordinal states. The previous analysis, actually, if you think every state, the difference from each one to the other is 1 point, and that's your utility, you can do a t-test, and it's clinically relevant. We've done this in other settings, a modified Rankin score. I haven't seen this done yet as a utility analysis of death, mechanical ventilation, and other things. You can do a Cox model on ordinal scales. It's not too different from a proportional odds model. And of course, you can dichotomize it. Death and non-death is a dichotomization, and then you can do a logistic regression model on it. You can do many of the other things we do in dichotomous tests. The Bayesian proportional odds modeling is pretty straightforward. If you have 23 states, for example, you have to be a little bit careful in the background alphas for the different values, and does your MCMC mix well, for example. It it reduces to a logistic regression model. There's a number of ways to model the background alphas in a proportional odds model. We could probably talk for an hour about the good and bad ways to do that. The interpretation of the odds ratio, I think, is a benefit of this. Covariates are relatively easy. Interactions of treatments, you can have a term for an interaction. That's nice. And of course, the Bayesian tied to a parametric model allows adaptations, response adaptive randomization, early success, early futility, futility rules that make clinical sense, all of those things, and we're involved in lots of adaptations within these trials. Most COVID-19 trials have adaptations, and you can see the urgency to have adaptations, and they make sense in the model. So for example, here's a graph we've presented, and what it shows on the left is an assumption on the 23 states of the Remap-Cap model. I'll say more about this later, but here we've taken the ICNARC study out of the UK for estimating the relative rates. That's an odds ratio of 1 is what we think the control will look like for a moderate population, as to those who will have 21 days free of organ support, to those 20% mortality and on. And then we can show for a proportional odds effect of 1.3, 1.5, and 1.7, how it has this constant proportional effect to better outcomes, where mortality goes, where other values go, and what an effect size means in a trial. And then we can show power. Many of these trials, for example, don't have a sample size. And it makes sense in this setting. We don't know what the sample size is. So we present power as a function of sample size, as a function of adaptation for the different effect sizes, as what's the probability of demonstrating superiority in a one-to-one trial. And we can present power as a function of odds ratio, and we can show what do those odds ratios mean in the trial. Just what I thought was a really interesting slide is, what does it mean to be ordinal as opposed to dichotomous? So suppose you had an effect size of 1.5. The red curve shows the power, and I'll focus on 500 patients. As a power, if we've got a dichotomous endpoint, say mortality, where it's 50-50, that's the power in the red setting of a 50% improvement of the odds ratio of 1.5, 500 patients. If instead we had a scale, and this is the light green here, where we broke up one of these endpoints. So it might be death is 50%, and then the other 50% is non-death. Suppose we had a clinically relevant breaking up of 25%, this 0.5 into a half, we get about a 7 point, maybe 6 or 7% increase in power by having that be for the same treatment effect broken up into two different values. If instead you broke up this 50-50 into a third, a third, a third, you get just under a 10% improvement in that circumstance for the same effect size by having the underlying endpoint broken up that way. If you broke up this 50% into multiple things, by the way, if you broke it up into 50, 45, 5, you get very little improvement in power. Essentially, 5% of your patients are providing extra value to you as a statistician to learn about, and you get about a 2% improvement in power. If you broke up that 50-50 into 25, 25, 25, and 25, you get about a 10%, 11% improvement in power in that circumstance by going from dichotomous to ordinal. Now, it doesn't help you if those are not clinically relevant values, But if you can do that, you can see that increase in power. So from a 50-50 to a 23-point ordinal scale, I probably should have shown this, we're expecting maybe a 10 to 15% improvement in power by that clinically relevant scale. By the way, the code used to do that simulation, to do Bayesian proportional odds modeling, there's code there for JAGS, there's code there for STAN. To do simulations, you can change the sample size, the underlying effect size. It's freely available. You can download it, of course, support free, but you can download it. And thanks to Lindsay Berry and Liz Lorenzi for doing that code and making that available and doing some of those simulations. So you're free to download it, test it out, run your own simulations, do adaptations, knock yourself out. All right, Tom.
A quick question that's come in, Scott. Is the odds ratio similar to the hazard ratio that uses the proportional Cox model?
It's a great question. They're not the same thing. You're going to get a little bit different. I think they're similar. If you were to take this and think of 23 values as censored outcomes, that it's time to event, but it can only happen at integer valued, almost like a visit schedule in a time to event where you get 23 visits and you find out when they fail, as opposed to a proportional odds model. We've done both of those and we get different answers. The interpretation is slightly different in that, but it is really very close. By the way, you can do that in facts. You can do a time to event where the event only happens on visits, and you can sort of create 8 visits to do the WHO scale in facts, to do Bayesian modeling of the time to event as opposed to proportional odds.
One last question before we move on to Ben, talking about the principal trial. Wouldn't the proportional odds model require a stronger assumption about the proportionality?
A stronger assumption than Cox model? Or than what? Than the controversial thing about a proportional odds model is, or statistically controversial, is that the effect is constant across the scale. So for example, an effect from 1 to 1.7 pushes all the outcomes proportionally to be better. That, in some settings, you may worry about that assumption. You might worry that it actually makes healthy people better and makes sick people worse. And you would see death increase, but dark blue get better. That would be a violation of the proportional odds, much like violations of the Cox proportional odds. For example, in stroke trials with clot clearing devices, we've seen clearly a violation of the proportional odds. So for the modified Rankin, it can be an issue in those trials. It does seem that early on, the devices might actually have been killing sicker people and helping healthier people. Here, that seems to be something that clinicians aren't worried about, that generally this is going to push you in good directions, but might be something we do sensitivity analyses over for sure.
And can I ask one more? Do you see time to improvement of 1 or 2 categories on the NIH scale? Is that considered as clinically relevant?
Yeah, boy. I think when they put this together, they thought of it as relevant. And I've had some of these discussions in the Remap-Cap trial. And this ICU-free days, or removing from organ support, removing people from mechanical ventilation to non-invasive ventilation, freeze up ventilators, it's an important step, they say, clinically. So they are interested in that. And of course, moving people out of the hospital. And I think perhaps the NIH thought maybe it's hard to collect the difference between 5 and 6. Maybe it's not important. Maybe you could lump 4, 5, and 6 together. I think you get a lot of people that might argue some of these are not as relevant as others. And maybe it's mechanical ventilation, hospitalization, non-hospitalization, and death. Maybe it's 4 outcomes. I'll leave that to the clinicians to describe, but there's a lot of discussion on it. And I think there's been a lot of people moving to this scale because in general, FDA has accepted it. WHO has accepted it. The NIH has accepted it. It now has tradition behind it.
So I think it's okay. We've got a couple of yeah. Can you handle a couple more questions?
Of course.
Or do you think we should leave? There may be a lot more on proportional odds, and we should defer it.
Maybe we'll leave that. I think it's an important discussion,
Okay.
for sure, and maybe leave that into our discussion.
Okay.
Let's get to Ben.
Yeah.
Okay, Tom, can you put up my slides?
Yep.
So I thank you, Scott, and Kurt, both for that introduction and the great discussion there on the endpoint. I want to spend my time talking about the principal trial. And this is a trial of treatments for coronavirus in a primary care setting. This trial was put together very quickly. And so I wanted to kind of walk through a lot of the considerations, the key things we had to think about as we designed this trial. Next slide. So here's a little bit of a timeline. In March 16th, we were contacted by colleagues at Oxford University asking for some help in a trial design in this primary care setting. And this is different from the settings that Scott's talked about in the hospital setting. A lot of the trials I've seen have been in hospital settings. This is kind of unique in the sense that we're interested in targeting the primary care setting before they get to the hospital. And their primary objective is to compare hydroxychloroquine versus usual care, an open label study. It's a pragmatic trial. So there's no placebo comparison. It's more just, is this going to be helpful in a pragmatic setting? They are targeting high-risk individuals, so older persons, either greater than 65 years old, or if you're older than 50 with some comorbidities, with suspected coronavirus symptoms. With suspected coronavirus infection. So what I mean by that is we're going to take swabs and find out who's positive and who's negative. But as we know from reading in the media, these tests are not entirely accurate, and there also are issues with supply of these tests. So we're going to treat patients regardless of what their test result is on coronavirus. If they have I think the great analogy is if it walks like a dog and barks like a dog, we're going to treat it like it's a dog. So the idea here, we're going to treat people who have the symptoms in the same way that we might actually implement this in practice. That's kind of the pragmatic nature of it. So their needs, what they wanted was a rapidly initiated trial with adaptive features. They wanted the ability to evaluate treatments quickly, so looking for early superiority or early futility, and in particular, the flexibility to add treatments. They needed a proposal for the funding source for the UKRI, and this was a non-competitive award, but basically they'd been solicited to put together this proposal. They needed an adaptive platform trial design, and they needed a data monitoring safety committee really quickly. And we put all this together, and I would say a record fashion. The first patient was randomized 2.5 weeks later. So if you want to know how quickly a platform trial can be put together, very quickly is the answer. And now there still are some moving parts of this, and I'll talk more about that as we go through this. Next slide. So I want to talk here's an overview of the key design considerations we had to think about and putting together this platform trial. So the first consideration is, what kind of endpoint are we going to go with? We're going to go with ordinal or a binary endpoint. Then the interim analyses, what's going to be the frequency of these? What's the timing? What are the rules going to be for early superiority or futility? Are we going to use responsive after randomization? And are we going to leverage the partial information at the interim analyses? So maybe we don't have complete follow-up, but maybe we have some other information for many of these individuals with the fast enrollment we expect. We also have considerations to think about with the control arm. The usual care arm may be replaced at some point during the study. And then we also have this idea of new arms entering the trials as we go forward. So we have this essentially staggered treatment arms with potential for a population drift. Now, maybe I'm not usually that worried about population drift in a lot of our platform trials, but this one seems like it could be a very plausible setting where we have a potential virus that's changing in the population. Maybe the prevalence of coronavirus positive goes up in the population. Maybe the hospitals, the healthcare system becomes more stressed and they change the criteria for hospital admission. So there's things that could change, even in a relatively short amount of time. There are things that could change here. So we need to think about that. And then we also need to think about what are the operating characteristics? What is the performance of our design? How does our adaptive algorithm perform? So we want to look at expensive simulations. We want to be able to then quickly transition to trial implementation and actually execute the trial. Next slide. So the primary endpoint, the principal investigator initially proposed a binary endpoint, hospitalization or death for suspected coronavirus within 28 days. And the first thing we did was we immediately steered him to an ordinal endpoint, as Scott's done a great job talking about the additional benefit we typically get from that. And so the ordinal endpoint that we identified early on was breaking this up into those who stay at home, those who get admitted to a hospital but don't require any sort of ventilation, and those who are ICU, and those who, the next level up would be those who require the ICU or ventilation, and number 4 would be death. And so, this is all, again, happening very quickly, but the initial phone call was, he was asking about the power gain we might get on this. And the initial response was, well, it might be a 5 to 10% increase in power, but we need to a quick simulation to really quantify this. Next slide. So Scott did a great job just talking about how the power is relative to the distribution that you're breaking up. And in this trial, we have approximately 1,500 patients that we're planning on randomizing per arm. And we expect about only 20% of those participants to undergo a hospital admission. And so, as Scott was talking about, when you add an ordinal endpoint of this nature, we're not touching the 80% who are staying home. We'd be breaking up the 20% who actually go to the hospital. Those additional 3 categories are breaking up the kind of hospital stay they have, whether or not they require mechanical ventilation or whether they die. And the simulations that we did, they only had about a 1% power gain for that particular setting because we weren't gaining much from breaking up the 20%. Now, if we had been able to break up the 80% who stayed at home, maybe less severe symptoms, or maybe they're better in 3 days versus 7 days, then we could have certainly gotten additional power from that. But the goal of the interventions and the goal of the trial here is to really keep patients alive and out of the hospitals, because that is what puts a strain on the healthcare system. And so we had to make a quick decision at that point, and we decided that the complexity of the ordinal endpoint, and I wouldn't say it's a lot of complexity, but there's a slightly more inferential complexity. It becomes a little bit more complex when we want to add a longitudinal component. I'll talk more about that. but also operational. We got to figure out how we're going to get that hospital, break up that hospital endpoint in the hospital setting and get that in a timely manner. And those complexities just weren't justified in our setting for 1% power gain. There were probably some different assumptions that we could assume to try to milk that power and get a little bit higher in terms of the power gain we get, but it just didn't seem like it fit this trial particularly well. So in general, I would certainly recommend an ordinal endpoint, and certainly worth exploring in these kinds of trials. This particular trial, that the verdict was, we're going to go with a binary endpoint, and that's going to allow us to leverage the partial information, which I'll talk about. You know, maybe we don't have 28 days worth of data, but maybe we have 7 to 14 days of data on some individual, and maybe we'll be able to harness that a little easier. Next slide. So the primary endpoint is 28 days hospitalization or death, yes, no. And the primary analysis is to a Bayesian model, and we're going to declare superiority of hydroxychloroquine to usual care if the posterior probability of superiority is greater than 0.98. This model is a Bayesian logistic regression model, and estimating the probability of hospitalization by treatment arm, adjusting for age and comorbidities. Next slide. And then we needed an adaptive algorithm, an adaptive design here. So in this setting, we expect very fast accrual. So we are expecting 250 participants a week. We're already at 150 randomized as of this week, and it's ramping up quickly. So this is certainly within the possibilities of what our expectations initially were. We scheduled the first interim analysis to occur after 100 participants, have the opportunity to complete 28 days of follow-up, and then subsequent weekly interim analyses. Now, it seems like a lot to have weekly interim analyses, but when you're accruing 250 patients a week, that's almost too long. And so we have to think about the frequency of interim analyses with respect to, relative to the expected accrual. And we have, each of these interim analyses, we have this Bayesian model to evaluate the arm-specific utility or superiority based on these Bayesian posterior probabilities. And the rules we're using for early superiority, if the treatment, the probability of superiority to usual care is greater than 0.999, so it's quite high, but when you're taking this many looks, we don't know how many looks there's going to be because this is just weekly. So it is quite high, but we've shown through the simulations that we're going to be able to identify the big effects early, and the moderate effects will take a little longer. The arm would stop for futility if the probability of meaningful benefit was quite low, whereas meaningful benefit is at least a 5 percentage point decrease. And the idea here is we want to control the type 1 error for each of these arms to be approximately 0.025 one-sided. That's going to require some simulation. But the initial design, at least getting the protocol and things through the ethics committees and to the UKRI and getting all that in place, we used facts for the initial simulations to calibrate this design in just the one-to-one setting where we're comparing hydroxychloroquine versus usual care, because that's the initial focus of the study. Next slide.
Just a quick, sorry, Ben, just a quick question. Meaningful benefit, is that a 5% decrease or a 0.05% decrease? Just clarify.
Oh, that's a good point. It's a 5 percentage point decrease, so that's a typo.
Yeah, thank you.
Mm-hmm.
Thank you, Andrew, for a good spot there.
So when you enroll this quickly, when we have an interim analysis, we're going to have many randomized subjects who lack the 28 days of follow-up. Normally, we think of 28 days as being a relatively short endpoint. But in this setting, we have a lot of outstanding data when we have an interim analysis. And even more so, some of those participants will have early hospitalizations. So maybe they only have 10 days of follow-up, but maybe they were hospitalized at day 4. And so the question is, how do we incorporate those without biasing the estimates? And you can't just count them without accounting for the the people who have had the same amount of follow-up who haven't been to the hospital. It would bias your estimates upward. And so the way we deal with this is we capture the early hospitalizations with a 7-day and a 14-day status. And this is done both through the daily diary entries of the trial and phone calls at 7 and 14 days. And the way it works is we get a 7-day status among those with the opportunity to complete 7 days, and we get a 14-day status among those with the opportunity to complete 14 days. And that allows us to build a longitudinal model for the probability of hospitalization by day 28 as being 1 minus a product of probability of surviving or making it without event to 7 days and making it to event without at 14 days, conditional on not having an event at 7 days, et cetera. So it's just this conditional probabilities, working your way through it and getting to the probability of a hospitalization by day 28. And what this will enable us to do then is if a person only has 10 days of follow-up, but they were hospitalized at day 3, that would get included in the analysis. And it allows us to provide a better estimate of what the treatment benefit is when we have this current snapshot of interim data. Next slide, please. The other thing we wanted is the ability to add treatment arms as the trial is ongoing. So we're starting with hydroxychloroquine, but we've already identified the next arm going in, which is azithromycin. And the arm after that is likely to be a combination of a different antibiotic, doxycycline, and hydroxychloroquine. There's safety issues with combining azithromycin and hydroxychloroquine, as many of you are probably aware. So there's various arms that are coming in, azithromycin being the next one that's scheduled to come in. But we need to have a plan for these. We need to have a pre-specified plan. If once that third arm comes in, that second intervention, then we're going to use response adaptive randomization to allocate subjects to the more promising therapies. And we're going to do that by fixing the control allocation. So the usual care allocation will be fixed. And then we have a choice whether, these are kind of things we had to think about and still are thinking about some in terms of what is the response adaptive randomization probability based on? Is it based on the probability of being the best intervention, or is it based on the probability of being superior to usual care? And if it was the latter, okay, what happens if you replace the usual care arm with a superior arm and you have a new standard of care? And how do you then, handle new treatments or treatments moving forward? Are you interested in non-inferiority relative to the new standard of care, or are we interested in the original usual care arm comparing to that? And if we're interested in the latter, which I think we really are, how do we compare to that original usual care arm if we're no longer randomizing participants to that arm? Next slide. So here's kind of a visual of what this could look like. So the usual care arm comes in with the black bar, and you have HCQ, hydroxychloroquine, in red. And those are the two that start the trial. Azithromycin comes in shortly after the trial begins. And so you have 3 arms, and then you have potentially a third intervention, drug C, which comes in later. But at some point, hydroxychloroquine could be deemed superior to usual care, in which case you would stop randomizing participants to the usual care arm. And the hydroxychloroquine could either be the new comparison, the new comparator that you're interested in, or it could serve as a bridge for the comparison, to make the bridge of the comparison of say, azithromycin in green versus the black versus the usual care. And I think the right comparison here is in a pandemic setting where we don't know whether we're going to have access to hydroxychloroquine in all parts of the world. It would be very, very useful to know if there's a second drug that maybe it's not as good as hydroxychloroquine, but maybe it's still better than nothing. Maybe it's still better than the usual care. So to go to the next slide, I'll give you a little visual of what I'm thinking. So on the left-hand side, we have a hydroxychloroquine distribution. These are posterior distributions of just made up data. But say you had an observed rate around 10% of hospitalization, whereas your hospitalization rate in the usual care arm was 20%. And so you've clearly shown that hydroxychloroquine is superior to usual care. And so you stop randomizing the usual care. But your azithromycin arm, the green one in between, it's somewhere around 15%. And now what we really want to know is, in a pandemic setting, clearly hydroxychloroquine is going to be the preferred drug, at least with the current available data, but is azithromycin is that superior to usual care. So if you don't have access to HCQ, is azithromycin better than giving nothing? And I think that's the right comparison. The way we're able to do that, so if we go back one slide, Tom. So the question is then, how do you compare azithromycin to usual care when you don't have those current randomizations, or how do you compare drug C to the usual care? And this is done by modeling this trajectory over time. And this can be done because we have this overlap. So if we go forward 2 slides, please. And so the idea here is we have a primary analysis where the comparison is each arm, each intervention versus the original usual care arm, even if you're no longer randomizing to that usual care arm. And you do this through modeling the drift over time. And this drift, okay, so we have this logistic regression model. It has treatment effects in it, it has covariates in it, and it has this function of time, this effect for time. And it's based on what's called a second order normal dynamic linear model, which creates Bayesian smoothing, hierarchical smoothing over these 2-week intervals. And as long as we have these overlap between the different arms, we're able to get an estimate of comparing future arms versus that original usual care arm. And I'm glossing over the fact that there's a longitudinal component of this for the interim analyses, but this would be the setup and the structure for the primary analysis when you have complete data. Next slide. So all adaptive decision rules, they need to be pre-specified. Now we're going to use simulations to evaluate the performance of this algorithm and the decisions that we arrive at. It's going to help us calibrate our design. Even though we put together a design within 2 weeks and we randomized the first patient, we're still simulating and calibrating, refining the design. And there's still potential to change things up until the first interim analysis. Once we have hit the timing for that trigger, the first interim analysis, the design is locked and we're going forward with that particular design. Next slide. And so we also, while we're doing that, we also have to simultaneously think about executing the trial. And so there's a Berry team that's actually moving forward with conducting the interim analyses. The emphasis, of course, is on timely data collection. That's critical, as well as having test data transfers. We got to figure out what data we're getting and what formats. We got to get the programming in place, the validation in place. We have an interim report generator. And this, again, this is done weekly, so we can't be doing this manually. There certainly is some manual effort, but we have to have some level of automation to this. And we have to clarify the communication channels for the interim analysis results, and we have to have firewalls between a design team and an execution team, because in a platform trial, we may have additional arms that want to come in. And so we have to be able to add appendices to the design and add simulations to characterize the performance of those designs. Next slide. So last slide, Tom. So I just want to thank Nick Berry, who's been instrumental in doing the bulk of the work and the heavy lifting in terms of the simulations, and then our execution team. So I've taken up more of my time. I'll stop more of my time than what I should have, but I'll stop there and I'll pass the baton back to Tom.
Well, I just want to ask you, one of the questions that came up to you, we've got quite a lot of questions, and they really fall into 2 camps, one on platform trials and one on ordinal endpoints, and we can have sort of 2, we'll break the Q&A up into those 2 topics. But there was just one thing you might like to, I like to think about commenting on, which is the idea of controlling type 1 error in a Bayesian design. It's not the probability of making a mistake of concluding that the drug works, it's the probability of asserting efficacy if there was no efficacy. And that preserving that is possibly lowering your Bayesian power.
So before you answer, Ben, I'll just let you know that Frank Harrell is listening.
That sounded like a Frank Harrell question. I don't know who asked the question.
It certainly was.
So yeah. So there's certainly some some philosophical debates about type 1 error and Bayesian designs, and that's inherently a frequentist concept. I think the goal here is we want to have a design that performs well no matter what the truth is. And so it's good and it's helpful to know how often we are going to get the right answer and how often we're going to get the right and the wrong answer. I think that's helpful here. We're not going to be overly picky about that 0.025, but I think in general that's a reasonable metric to think of a frequentist type 1 error of a Bayesian design.
And to a brief a question, sort of follow-up question, but from Tobias, the 2.5% threshold for efficacy, is that planned to be a seamless phase 2-3 platform? Is there acceptance for approval being discussed with regulators?
So there is no regulatory involvement here. I mean, so these are already drugs that are available, mm-hmm. and so there's no regulatory involvement. It would be more making this information known to the public and conveying that information. So, you know, I imagine the regulatory agencies in each of these different countries would want to have some sort of input on the design and the results and maybe change the label, but we're not concerned with the label. This is an academic trial trying to find the best way to treat coronavirus with existing therapies.
Okay, thank you. We'll get, we're running behind time. We'll get straight to Scott on Remap, and then we can go to the Q&A. I need to give up presenting. Here we go. So it should be available for you, Scott.
All right. Sound check again. You can hear me, Tom?
Yes, certainly can.
Okay. So I'm going to talk specifically about the Remap-Cap trial. All right. So the Remap-Cap trial was, you see this list of sponsors and people involved. I'm a little bit hesitant to list people involved because this has been such an incredibly large and worldwide effort. So I'll try to give a little bit of history that sort of puts in place what this is. Remap-Cap was originally, it was a reaction to the H1N1 and really a poor global reaction to the clinical trial seen in that reinforced by the poor reaction of the scientific community to the Ebola outbreak. So there were years of this, and there were a number of intensivists that said, boy, we need to get into this platform trial effort. We need to do something to be ready for the next pandemic. So Remap-Cap was funded by the European Union, and there were multiple efforts in that. Ben had an influenza trial, a nice influenza trial that came out of that. One of the work packages was Remap-Cap. It was creating a clinical trial in community-acquired pneumonia that would enroll in what I'll call peacetime, interpandemics. It would enroll community-acquired pneumonia. This is an ongoing issue in intensive care units, and we would learn about the way to treat community-acquired pneumonia. Meanwhile, it had what I'll call a sleeping strata, that if there's a pandemic, pandemics will most likely end up in intensive care units, and the trial is already enrolling in intensive care units. The infrastructure is there, the protocol is there. The protocol already has a strata for pandemic. It was just called a sleeping pandemic strata. We don't know what it is. We actually were expecting it would be influenza, but whatever it was, we just fill in the name and it's already ready to go. The trial's enrolling in sites and all of that. So that happened, and I think it was February 29th this year that the trial declared a pandemic. I may be off on that date. And the sleeping strata became a protocol. It became a stratification, and the trial's now enrolling COVID-19. earlier in February, the places that had sites didn't have patients. We didn't have patients in Wuhan, and there wasn't the ability to enroll. So we were sort of sitting and waiting, and then it was declared there were patients. At the time, there were sites in Europe, Australia, New Zealand, and Canada. And there was something in the order of about 40 to 50 sites. The trial actually had been a long time in development from about 2016 on. Had this been COVID-20, we would have had many more sites and more well-oiled machine. So it was a bit of a hurried effort to get this. And now this has been extended to the US. It took a long time, but is now enrolling in Pittsburgh. You can go to the site and see they've enrolled 269 patients as of this morning. You can see the ticker. The protocols are online. There's a great deal of public material for the trial. So it's a very open trial in what it is. By the way, Remap is randomized. So the trial is randomized. It's embedded in so that some places do this as a part of care. At Pittsburgh, for example, this is done by video consent. The EHR is the database, so there's not a separate database. An order sheet comes out. It says, do you want to randomize the patient? It gets consent. The patient's randomized. It says, okay, do this to the patient. And it's all embedded within the healthcare. It's multifactorial. This isn't a trial where you do A, B, or C. It's a trial where you do a number of things to each patient. It's adaptive, and it's a platform trial, is the Remap. So it has now, since the COVID has become the stratification of the pandemic, it's being extended. I believe it's at over 100 sites now, and within a few weeks to a month, there'll be over 200 sites enrolling globally. It's one of the nice things about this trial that if the disease wanes in one place and waxes in another, patients are there being enrolled globally. There's discussions of extending this to South America, to the Middle East, though it's not clear the US isn't the place to be enrolling patients. So it's globally positioned to it. So what does the trial look like? The trial is enrolling suspected or proven. By the way, it's still enrolling non-COVID patients in community-acquired pneumonia and investigating multiple therapies for CAP. It classifies patients into severe and moderate. It doesn't enroll mild. It's working, it's co-enrolling patients, it's working with other trials where mild may advance to moderate and then they become randomized within Remap-Cap. The way it's structured is that within each disease state, there are domains. Each domain is a set of mutually exclusive interventions. So for example, an antiviral domain is, one intervention is no antiviral, one is HCQ, hydroxychloroquine, one is Kaletra, and one is Kaletra and HCQ. So there are 4 mutually exclusive things. Every patient gets exactly one of those. And then there's a immune modulation, there's a steroid domain, there are multiple domains in the trial, and they're different for the different state of disease. Some of them overlap, where we give the same thing to patients in both statuses. So a patient is randomized to a regimen, which is one intervention from every domain. So they get assigned an antiviral, they get assigned an immune modulation, which by the way, might be no immune modulation. There might be multiple immune modulation states, there might be a steroid state, a macrolide state, which is azithromycin, anticoagulation. So they get an assignment of each one of them. So this is the multifactorial aspect of this trial, which dominates this, is that every patient gets lots of things. So you might be a control in antiviral, because you don't get one, where you get a immune modulation therapy. So each patient is randomized a regimen. Some regimens they're not eligible for, and they're in the non-eligible state for that, but they get randomized to everything else. A patient can progress from moderate to severe. I don't necessarily want to get into this issue too much. Jack was already asking a little bit about this, where they could then become eligible for domains in severe, and then they're randomized to them. If, for example, a mechanical ventilation, how do you set the vent? They may become eligible for that domain that's only for severe, that we then randomize them to that, or reveal the randomization that was not revealed until they moved into that state. And the protocol defines how that's done and how we deal with that. The endpoint is, as I described, this ICU free days endpoint. So this 23-point ordinal scale, regardless of disease state. So that's the structure of what happens to a patient in the trial and the data that are then collected for that patient. Interim analyses are done periodically. That's what it says in the protocol. They're done periodically. It'll be a function of enrollment and determined by individuals blinded to the data. The current plan is weekly. We'll be doing interims weekly during the course of the trial. Depending on enrollment, it might then do every other week. It may move to monthly, depending on the status of the trial. At any time in the trial, we can add things sort of outside in. It's moving in. You can add interventions. In the antiviral domain, we might add a new antiviral. Remdesivir or another antiviral gets put into an existing domain. The trial already knows what to do with it. The rules are all set. There may be specific things for the appendix of that go into it, but otherwise it just keeps running and it enters into the response adaptive randomization. You can add a domain, and I'll describe a little bit more about that. That's being done almost daily in the trial, almost weekly. So it has to incorporate the fact that we're learning an incredible amount about this disease, and the trial needs to be able to add that in the trial as quickly as possible. We can also add states. Actually, the moderate state is something that is there, but was added after the fact. Severe was enrolling, and now it's adding moderate. It's a bit of a US expansion that then moves globally. That state will then be done globally throughout the Remap-Cap trial. What happens from the data itself is that there are prospective rules that we can drop interventions for futility. So in some of these domains don't have a control. Everything is considered an active therapy. If there's a control, we can drop an intervention because it's futile compared to its control, or we can drop it because another active arm is better. It's inferior. That would be public disclosure. We would say agent X is not as good as agent Y. It's been dropped from Remap-Cap. By the way, it might be kept in if, in fact, drug supply is an issue. So the steering committee might leave it in under different circumstances. So public disclosure happens on a rolling basis in the trial. You can have an intervention win a domain. It's better than everything else. It gets named superior. All patients get assigned to it. So it's deterministic that all patients get it during the course of the trial. Again, depending on regional differences or drug supply, we might not drop things if needed in the pandemic. That's a steering committee decision. Response adaptive randomization. So the response adaptive randomization is done across regimens, not individually in domains, because a number of these have interaction terms. antivirals and steroids, antivirals and immune modulation may have interaction. So we calculate the probability that every regimen in the trial is the best way to treat a patient, and the randomization is proportional to that. There's a few constraints on it, but essentially it's proportional to that in the trial. And so we update it weekly to give new randomization. So we're trying to treat patients during the trial as well as we can. We're also trying to learn more efficiently because we give things that are better together and separately during the trial to learn more effectively how to treat.
Is there any sort of allowance for geographic area that comorbidities may be different in Europe compared to Africa?
Yes. So that's a great question. So in the model, which is a large Bayesian proportional odds model, with the 23 states as the cumulative proportional odds model, site is a factor in the model. Site is nested within country. So there's a hierarchical effect that UK sites are likely more similar than Australia sites or US sites. But every site has an effect, and it's nested within a hierarchical model of country, which is nested within global as a 3-level hierarchical model for site. Those are additive effects. We, at this point, don't allow a treatment to work one place but not another. So everything has a proportional effect across them, but we have additive effects by site and by country. I will say that we're adding convalescent plasma, and there have been lots of discussions that convalescent plasma may vary by region in how it's done and the plasma received. So we have a sensitivity analysis for convalescent plasma by region, as that's a concern in the trial. There's a time machine, as Ben described in his trial, so I won't go into that every month we allow an additive effect due to time. And it's very likely that in background stuff that Remap-Cap doesn't worry about, it does worry about things like azithromycin and steroids and vent and other things. There may be other stuff being done. Patients may change, so we allow that to change monthly over the course of the trial. Age is a factor in the model, as it was in the original model. We then have a factor for each intervention, and interactions within the design team specifies we want them in the model. Some don't have interactions. There is one set that is now enrolling where there was a publication in JAMA that there may be an interaction between interferon and steroids. And it might be negative. And so the trial started, I'll say non-objective, it's sort of a subjective based on that empirical information, where the response adaptive randomization starts where it discounts that. It doesn't give many patients to the interaction because it starts with a 75% chance that that's a negative interaction. And that starts from the get-go. So we're trying to incorporate global findings in the trial where necessary. The current state for Saville is an antiviral domain, and I described that to you. There's an immune modulation, which is non-interferon, anakinra, or IL-6. There are 2 IL-6 enrolling, and that's tocilizumab or sarilizumab. By the way, there are global differences. Those are enrolling only in the UK. There's only drug supply available for the UK, so those are grayed out at other regions, and that's fine. We learn about the contrast between the others in the other regions, and we're learning about IL-6 in the UK. That may happen in other circumstances as well. Steroids, yes and no. It's the most commonly used drug in this disease, and so we need to learn, is it good and how do you give it? As well as azithromycin macrolide when an antibiotic is given with azithromycin. We are adding an anticoagulation domain, which is heparin or not. By the way, that's a domain that they said in the primary analysis, we don't want that to have an interaction with anything else. It essentially comes for free in the model. It's 1 or 2 parameters in the model to learn whether heparin, a blood thinner, an anticoagulant, sorry if I'm butchering the clinical parts of this, but that this is we get that for free in the setting. So we're learning about multiple therapies simultaneously in the same patient. Vitamin C has no interactions in the model. Does it work? Yes or no? ACE2 inhibitors, it's a dash line around that. That's not likely to come in this week, but very soon. And that by itself is a 3x2 factorial on different therapies. That's not in there yet. These other 2 should be in there soon. So again, randomizing over 6 things. The moderate has started later. It's running with HCQ, yes or no, and then steroid, yes or no. And then we've got a few in there. I won't say what they are, but those are likely experimental or repurposed pharmaceutical drugs where pharma is partnering with Remap-Cap coming into the trial. Some of these feel like comparative effectiveness, and some of them are experimental. Though in a novel disease, in some way, everything is experimental. I showed before some of the simulation type things. So this graph is actually, if we bring in something like heparin, this is with response adaptive randomization, assuming we do interims at 200, 400, 600 through 1,000 with different effect sizes. How quickly could we determine superiority in the trial? We have similar graphs for futility. We have graphs for 3-to-1 domains, 3x2 domains. So the simulation document of this is really quite large in looking at the different types of domains and interactions across domains. Oops. So Tom, I'm done with that.
So thank you very much, Scott. I think what I will do is we'll stop the recording here for the presentation part. I will resume the recording for the Q&A, but we won't be making that public, but we will sort of type up a sort of summary of the Q&A if it looks interesting. Okay.